So thank you very much for this opportunity to actually spend the next 10-15 minutes presenting some work we've done in these preclinical studies, developing the idea of gene therapy that we proposed a couple of years ago. And this is the experience of Great Ormond Street Hospital. I think it's important to highlight that the majority of the experimental work that I'm going to be presenting has been undertaken by these two ladies who are in the audience, so you can have a direct conversation with them at the break if you want, Dr. Ying Hong and Dr. Marina Casimir. So I think the majority of the actual talks that we had so far have actually highlighted all the challenges we all have in terms of making treatment decisions for our patients. So corticosteroids, I think, still have a role. Other more traditional therapies that we have used for other autoinflammatory diseases, such as colchicine or anakinra, do not appear to be effective. A number of other conventional DMARDs that we used for vasculitides over the years, such as cyclophosphamide, are not commonly used and do not seem to be effective for our patients. And anti-TNF-alpha appears to be effective for the vasculitis autoinflammatory phenotype, perhaps less evidence with respect to the efficacy of these treatments for immunodeficiency aspects or the bone marrow failure, other hematological manifestations we discussed today. And I think we're all experiencing the unusual few patients that perhaps, despite the fact that they are on anti-TNF-alpha, over time perhaps escape efficacy. We had a patient ourselves who, despite the fact that he was actually anti-TNF-alpha, acquired an acute lacunar stroke. Allogeneic hematopoietic stem cell transplantation, of course, has been proposed, but even the transplanters within the audience have said before that it may be a curative option, but there are significant caveats with respect to the morbidity and morbidity associated with this approach. So why think about gene therapy? With taking all these caveats into account and the challenges about treatment, I think we're all looking for a possibility of really curing this disease, and I think this is what our patients and our families are telling us as well. Why would this be an approach to take for this disease? Well, I think the first key point is because it has an autosomal recessive nature, so therefore the theory that this is due to a loss of function mutation, and therefore by delivering a coding, normally coding really gene, you may be restoring the activity and restoring protein levels, and therefore this may be curing the disease. The success of allogeneic hematopoietic stem cell transplantation, of course, highlights that CD34 cells may be a suitable target for us for gene therapy approaches. I think we heard the healthy arguments about endothelial cells expressing or not expressing DADA2. I think even if there are low expression levels in the endothelium, the fact that DADA2 is an extracellular enzyme, restoring the levels to normal, healthy levels could really possibly reverse the phenotype, even considering the endothelial dysfunction of the disease. And we, of course, have experience with treating DADA1 SCID with this approach of gene therapy. So on the background of this, we actually hypothesized, together with our collaborators, Professor Adrian Thrasher and Bobby Gaspar, that using gene transfer strategies, we could possibly correct, starting with some in vitro studies, the defects associated with DADA2. And we had a number of different objectives when we started this study, and I will take you through these objectives step by step. So very firstly, we wanted a stable knockout cell line, so we can actually define the immunophenotype of this cell line in terms not only of the monocytic function, but the interaction with the endothelial cells, so we could then have some robust readouts of what we were trying to correct with the gene therapy approaches. So to do this, we used the CRISPR-Cas9 approach in THP1 cells, and then from the transfected cell pool, clones that were surviving were FAC sorted and then further expanded. And as you can see in the clone analysis sequencing here, these clones were not expressing DADA2. This was also confirmed using a number of Western blots, and we got 2 clones where we did immunoblot there, and you can see there's absence of protein. Also, we confirmed that in our DADA2 CRISPR-Cas9 knockout cells, we actually had no DADA2 enzyme activity, whilst the wild-type THP1 cells were continuing to healthily demonstrate DADA2 enzyme activity. Then we went on to actually study the pro-inflammatory phenotype of this DADA2 knockout cell line. So we took the THP1 cells, following the CRISPR-Cas9 transfection, and then we differentiated the cells into macrophages and then polarized them into an M1 or M2 phenotype using different stimuli over time, and chose some markers. I'm aware in the literature there's a number of different markers one can use, but these are the ones we used. We, in the initial phase, did PCR, and we looked at the different cell populations. And as you can see in this graph here, the M1 macrophages from the knockout cell line were actually expressing significant levels of TNF-alpha. This also applied to interleukin-1 beta. Again, you can see the knockout cell line compared to the actual wild-type cells, STAT1, and also CXCL10, so a number of pro-inflammatory molecules. We then also wanted to confirm that these knockout cells were actually releasing increased amount of TNF-alpha in the culture supernatant. So as you can see here, when we took the DADA2 knockout THP1 cells, you could see a significant upregulation in the M1 macrophages of production of TNF-alpha compared to our wild-type cells. How about the anti-inflammatory phenotype? Well, it's been speculated through a number of publications and presentations that in actual fact, one of the features of DADA2 is the lack of induction or the apoptosis of M2 cells. So what we've done is we actually looked at the ratio of M1 or versus M2 cells. So you can see the calculated ratio here, M2 versus M1. And what we were seeing in the wild-type THP1 cells was actually there was a significant upregulation compensatory of M2 cells compared to the M1 cells, whilst we were not seeing this compensatory mechanism activated in the knockout cell line. Is this comparable to the immunophenotype of macrophages one can see in DADA2 patients? Well, we did some preliminary experiments, and indeed it appeared to be the case. I'm just showing you TNF-alpha and also CXCL10, that actually we're getting the same really pattern in macrophages derived from our patients that we were seeing in the cell line. The same applied for TNF-alpha production, just about reaching statistical significance there. We need some more endpoints. And the same applies for the M2-M1 ratio that we were seeing in the cell line. This is resembling the immunophenotype of macrophages derived from patients with DADA2. So that really was the first step, some robust readouts and a stable cell line that we can now use to proceed with the gene therapy strategies approaches. The next step was actually to develop some specific lentiviruses that can deliver the healthy gene. And we did this with collaboration with Professor Adrian Thrasher's group. So what we used was actually a self-inactivating lentivirus with a vector backbone that was used in ADA1 skin disease already, which we then optimized for a coding ADA2 cDNA and eGFP under the transcriptional control of a promoter called EFS. And we also obviously had a self-inactivating viral vector that did not have the protein, just had eGFP. We then wanted to look at the THP1 cell line that we had developed and also some patient immune cells in our preliminary experiments and see can we actually restore protein expression or ADA2 enzyme activity, rescue the immunophenotype of these cells, and actually could we reverse the interaction with the endothelial cells. So we very firstly set off with different multiplicity of infections. We opted for 7.5 and 10, I'm showing you the results of 10, to establish the transduction efficiency in our THP1 cells. And as you can see, this varied between 20 to 30%, shown in two different ways in flow cytometry. We then did Western blot to actually look at the ADA2 protein expression. This is reminding you the wild-type cell line, expressing the protein, the knockout not expressing the protein. And when you actually transduce the cells with the virus expressing the protein, you can actually see we're getting a very healthy band, being able to restore the protein expression. And these are relevant controls just showing that the empty vector wasn't really doing anything. So for the very first time, we are actually showing that with this lentivirus encoding ADA2, we are able to restore protein expression in this knockout cell line. We also then wanted to confirm that this was expressed in terms of restoring ADA2 enzyme activity as well, not just the protein expression. So what I've summarized here for you is the ADA2 enzyme activity in wild-type THP1 cells. And as you can see in the uncorrected cells, we had hardly enzyme activity, whilst when the cells get transduced, you can see a healthy enzyme activity restoration. If anything, we're titrating to try and downregulate this a bit, so it's not so high. We also wanted to explore to see if we can down-regulate, suppress TNF-alpha release from these cells. And again, as you can see here, this was the TNF-alpha release from wild-type cells, uncorrected significant release of TNF-alpha, which we're able with transduction to bring down to almost the levels of healthy wild-type cells. We then moved on to some preliminary experiments using macrophages from our patients. So these are experiments at the very beginning, but you can see, again, we started with transduction efficiency establishment in our patient cells. We're using a higher MOI for these cells, and again, you can see the actual transduction efficiency varies between 20 to 30%. A very interesting observation you made that actually the patient cells seem to transduce to a higher level percentage than the actual healthy cells, as if they're actually very thirsty to uptake the virus that expresses the protein. We then, for the first time, actually used a flow cytometry antibody that's actually now available, and I think very interestingly can be used to try and answer some of the questions we've heard about this morning in terms of actually quantifying the protein expression levels for some of our patients. You can see the lower expression levels for DADA2 patients versus a healthy donor. So we use this as a readout for our patient cells following the transduction experiments, because obviously doing a blood you need a lot of cells. Well, this was much easier an approach to use. So this is the expression for healthy donor macrophages. These are our uncorrected patients, and as you can see, following transduction, again, we're restoring the DADA2 expression in our patients to healthy levels. And when you actually deliver the GFP DADA2 coding viral vector to healthy donors, you just increase the expression slightly more. Does this reduce TNF-alpha release from our patient cells? Indeed, that seems to be the case in our preliminary experiments. So healthy donor levels, TNF-alpha release from M1 macrophages, DADA2 uncorrected, and again, slight down regulation for the patient cells that were transduced effectively with this lentivirus. How about the apoptosis of M2 cells? Very crude assay, just looking at PI of M2 macrophages that have been cultured for about 7 days and then transduced with the lentivirus. As you can see in the DADA2 uncorrected cells, these are quite high, the percentage of apoptotic cells, which we're managing to bring down in corrected cells, gene-corrected cells, compared to levels that we are seeing in the healthy donors. And lastly, thinking about endothelial really dysfunction, very crude experiment of taking culture supernatant for macrophages from either gene-corrected or uncorrected DADA2 patient cells or healthy donors, and looking at expression of E-selectin on HUVEC. So you can see that significantly up in uncorrected cells, and we're managing to downregulate that to levels of healthy donors with transduction with this lentivirus encoding DADA2. So I hope in summary that I've convinced you we have a cell line that's stable. We have established some immunological phenotypic features that we can use as a readouts, but I take all the conversations for reflection that we had today about additional readouts we can develop in the cell line. We have developed lentiviruses that encode the protein, and we have been able to show in these experiments in both cell lines and actual patient-derived cells that we can restore protein activity and enzyme activity. We can rescue partially the immunophenotype, a lot of other readouts to obviously explore, and we're on the case for that. Reduce the inflammatory interaction within the endothelial cells. And what are our next steps? What we're thinking about is obviously the CD34 cells and working with patient cells if possible. So if you do have any available, we very much would like to talk to you, and I'd welcome the opportunity to collaborate with you. Obviously, thinking about animal models, we need to establish not only the efficacy but the safety of this approach, and we have some ideas how to take this forward. And I hope I've convinced you that obviously these are experiments at the beginning, ongoing hard work, but there may be a real possibility of a cure of DADA2, but we need international collaboration to actually deliver this. Thank you.